Today we are going over biochemical. Biochemical. So, stains tell you what? In morphology. Very good. So, but biochemical tests, they tell you what the bacteria can do. Okay, what the bacteria can do. Um, so this is the whole point of this lab. So we're moving forward from appearances based ID to now metabolic based ID. So why do we need biochemical tests? Well, we need biochemicals because in the earlier labs, you use staining to identify purple, pink, negative, positive, the shape, which is all the morphology, the arrangements, and if there's capsules, pairs, endospores, flagella, stuff like that. So now, um, these all test the metabolic abilities of those bacterias. So, meaning it's asking what can, where was I at? This bacterium breakdown, does it use ferment? Does it digest? Or does it produce? So the logic here is that each bacterial species has different DNA, okay? And that different DNA means that they have different enzymes, and that different enzyme means that they have different metabolism, and those different metabolisms give different chemical reactions. And those different chemical reactions are from our biochemical results. So you guys did all these, right? With your unknowns. So this is why biochemicals can help identify the bacteria, because they're looking at can this bacterium ferment glucose? Can it make acid? Does it, make gas? Does it use does it have a carbon source? What carbon source does it use? So all of this is what it's asking. So mostly the lab focuses. So the lab focuses on enterics, with the exception of Pseudomonas, because remember that's not an enteric, not an enteric. But for the most part, all of them are. So the family it belongs to is the Enterobacteriaceae. Did I spell that right? No. And what they all have in common is that they're all gram negative. They're all rod or bacilli. And then they're all facultative anaerobes. These should have been givens when you guys did your lab. So why, let's see. So the tests we're going to focus on are, we have the oxidase test. We have the citrate utilization test. We have the urease test. We have EMB agar. We have the MAC agar. We have the SIM. We have fermentation, which I'm not sure. Some people brought it to my attention that she guys didn't give you a packet that she gave our class. So for our class, we had to learn it as carbohydrate fermentation. But for you guys, I don't know. When I asked her, she said to use carbohydrate fermentation, but someone brought it to my attention that you guys did not learn that. So whatever you guys learned for that test, that's the name of it. Oh, the bacillation and the optic. Okay, so she's taking those bacteria, the bacteria antibiotic ones out. Okay. But you do need to know your alphas, betas, and gamma. Yeah. Okay. And then the fermentation, that breaks up into two, which is the BCP glucose and BCP lactose. Okay. So what is glucose? What kind of sugar? Simple sugar. Simple sugar. Very good. We have examples of what sugar, simple sugar is. Anybody know?
Reaching, but monosaccharide?
Yeah, monosaccharide, but real life. So we have our blood sugar. We have honey. We have fruit. Well, let's not use fruit yet. So another form of simple sugar is fructose. That's also simple sugar. And that one's for fruit. We also have sucrose. Is sucrose a simple sugar? Does anybody know?
I thought it was a complex sugar.
Of what? It's a mixture of glucose and fructose. An example of that could be table sugar and candy. And then we also have lactose. Does anybody know what if lactose is a simple sugar? No, that is glucose and galactose. We're not going to go into galactose because you'll need to learn that later on in nursing, not for this. But an example of lactose is sugar in dairy products. So like milk, yogurt, etc. So for this one, we're gonna talk about let's talk about fermentation. Okay. So fermentation, actually, Yep, let's go into it.
Why did you go into all the sugars the way you did? Not that I mind, I'm just curious where this plays in.
I think there's a question of what if like glucose is a simple sugar or not, and if lactose is a simple sugar or not, and it's not. Well, lactose is not. So I just wanted to break it up, and then a lot of people, well, at least last semester, a lot of people got confused like on the sugars, and wanted to know, well, how come it doesn't test for sucrose? Because I think one of them tests for sucrose too, which is glucose and fructose together.
Fructose and lactose are complex.
Complex, yeah. Okay, so where was I at? Fermentation. Fermentation. So fermentation asks, did the bacteria ferment the sugar? It's also asking, did acid form? And then lastly, it's asking, did gas form? These are the three things you're looking at during a fermentation test. Okay, so first we start with, let's not start with that yet. So we have two different media types within the fermentation test. We have selective and we have differential medias. So selective chooses who can grow. It contains ingredients that inhibits, so ingredients that inhibit certain bacterias. And then differential shows differences between organisms that do grow. So selective and differential, they don't just ask, can you grow here? And what they actually ask is what, so they don't ask, can you grow here? But what they do ask is what do you do once you grow here? So what do you do once you grow here?
Like test questions are not gonna be asked, you said?
No. So this one is don't. It's not just asking like, can you grow here? It's asking, what do you do once you grow here? So it's gonna, yeah. Okay, we're gonna talk about our pH indicators really fast. So we have pH indicators. And we have three. So our first one is gonna be phenol red. And below pH 6.8 is gonna equal yellow. pH of 6.8 to 7.4 is gonna be red. And then pH of 7.4 plus is gonna equal pink or magenta.
Are these 1, 2, 3, or is this 1A, 1B, 1C?
No, so this is all phenol red. 2. Which one? So 2. Donut. I'll come back to that. Okay, so 2. We have BCP, which stands for, I don't know if she's gonna have the whole name or the initial, but the whole name, so you guys know, you guys can't say I didn't tell you, is bromocresol purple. Okay, so below pH 6.8 equals yellow, and pH 6.8 plus equals purple. And then we have 3, which is BTB. Again, I don't know if she's gonna have the initials or not, so it's bromothymol blue. And below pH 6.0 equals yellow. pH 6.1 to 7.5 equals green. And pH 7.5 plus equals blue. Okay, so going back to 1, yellow what does yellow mean? Very good. So did that ferment or non-ferment?
Why do you draw that conclusion from that? So because the pH is below, because the pH was 6.8, it's yellow, what did it start as? So what color? We're saying if it's yellow, if it's red, if it's pink, what is the control? Like what is?
I think it's red because it's phenol red.
And then if it turns yellow, that means that it's acidic, so that means it fermented.
This one's purple and this one's blue. So the red, is it acidic, alkaline? Normal, neutral. No, because basic is what? No. Basic is also alkaline, yes. So don't use basic because she'll think that's alkaline, that's wrong.
Like them, so it will be like, red, if it's like yellow.
If it's yellow, then what does that tell you? If it's red, Neutral. And if it's pink or magenta,
I mean, basic.
basic, which is also alkaline. And that means, does anybody know? It means no sugar, but something happened, right? Because it turned green. I mean, it turned pink. So something had to happen, right? So nothing was fermented, but it had proteins. And they broke them down.
Would that be the same as like nature or no?
Yeah, but with proteins. With proteins. Not sugar. Not sugar. So what is the yellow tells you what? That there's sugar. The sugar was fermented. The pink tells you that there's no sugar. Okay, no sugar, but there's proteins in there, and some sort of enzyme reaction happened, which broke the proteins down. That's what it tells you.
And this is all taking place in a broth?
Okay, now we got BCB, BCP.
For one, like I was saying, what testing we like correlate to find this and all this?
This whole thing is your biochemical tests,
Yeah, but which,
your tubes, your lactose and glucose.
This is the fermentation test though, right?
Mhm.
So when you're asking, when she's asking which one would it be safe to say, which test would be the fermentation test?
but then when we're talking about,
No, she's gonna have all the tubes on there. I don't know about Hewitt, but she's gonna have all the tubes on there, and she's gonna ask you which one's which.
how red is the MSA plate, right? But that's why I'm.
Right. But if she has a red broth. Mhm.
I don't recall ever not having a red. So the broth could be any color.
It could be yellow, red, or pink if it's the phenol red.
That's just the one we saw. It's not good. That's why I'm kind of like confused.
I know, but these might be the okay, so the red and the pink might be the written questions. Yes. The actual labs that you see are gonna be the ones you guys did in lab. So they're all gonna be yellow because they all were yellow except for the ones that were lactose. And what color was that? Purple. But these all might be written questions, so you guys still need to know this. Plus, these were on your guys' lab notes, on your lab sheets. So all this information's on there. So you guys, it's all free game. You know how she loves her free game. So she loves, yeah. So, and I know you guys don't know I know you guys don't read because you guys don't be reading my emails that I send out. So you guys don't know how to read and you guys don't know how to write. So I am helping you guys, all right? This is something you guys need to pay attention because I know damn sure you guys are gonna go home and not take a look at that lab stuff that you guys have done. You guys completed it and you guys are done with it. You guys moved on. I know you guys because I was once you guys, okay? So I'm breaking it down. I'm teaching you guys my mistakes so you guys don't get dock points on your guys' lab practical like I did. So BCP. All right, yellow, what does that mean? Acidic, very good. What did it ferment or no? What did it ferment? Which sugar? Which one? It's purple, so what does that say? Lactose, very good.
So that's where the dairy products came in. It's because lactose is a type of sugar.
Yeah. And purple, neutral. So it's no acid.
So the first one we're talking about is single.
Yes.
In particular, which one?
Which one? You tell me.
Or are we testing for both? We're doing glucose and glucose?
So which one is it? If we already have. If BCP does what does phenol red do?
I'm confused too.
We always push it on.
Yeah, so what's the other one?
simple sugars are up here. The glucose is and the fructose. So then fructose, we kind of don't need to know. Good to know, but that's okay.
It's good to know, but it's not the one of the labs. So remember I said we're only focusing on labs.
But then we also have to so just in case somebody word from it. That's what I'm getting. Cause I know, I remember lab, but then
Okay, so then for this one, this is the lab you did, right? You did this lab. So what's the sugar?
see, I don't remember.
No, don't look at it. So we tested for two sugars, right? One was lactose, and one was glucose. Right.
So then that's your answer.
So if it's purple, we're gonna know that it was lactose, and if it was red or yellow, it's gonna be glucose.
Well, it could be yellow on the lactose too.
It could be yellow on the lactose too, yeah.
If it's purple, then you should know it's gonna be lactose. There's no purple in any other ones.
How would we know if we're testing for a simple sugar or a complex sugar? Technically, we could have any of the tests.
I don't think she would test. I think she's gonna have
Or maybe for the
I
people-
don't so I don't think she would have both a lactose and a glucose that are both yellow. I think she would have one that's purple. Or that's why I also make sure you guys know the red or pink, because she might switch it, because there's no way she can have both yellow.
Like maybe not first.
Oh yeah, that's what the worksheets are. Yeah. Let me just get through the lecture so you guys can actually have it. Yes.
So the exam, there's gonna be like, is it gonna be like 2 or 3? There are gonna.
All of them.
All of them?
All the tests you guys did.
So one of them will have to be purple, and then the other one will have to be green or blue, because they all could appear yellow, right?
Right.
Because that's how we need to like differentiate them.
But they're all gonna be in twos, right?
So the yellow, again, is acid. Honestly, I don't even think this one's on there. It's on the paper, so that one might be for game, but I don't think. What I do with my sheets. Oh. It is.
Can I see if mine matches yours? Because I see nothing with the phenol red. It was just either BCP broth black or BCP broth blue.
This one I had.
This is good information.
This one, which is like sweet diet notifications. Wasn't even on board.
Or is it a little bit different?
This one's different.
Okay.
Okay, so yellow acid, that means it fermented. Green. What would that say? What do you think? Looking at the pH, neutral. Very good. So no reaction. And blue would be what?
Alkaline?
Alkaline. That means something broke down. Okay, so I messed up, sorry. So the phenol red. This one. Or, okay, since you guys are so worried on left. So the only one you need to worry about for lactose and glucose is this one. Lactose and glucose is this one.
Yeah. but
Is
that
this what she was talking about?
Somebody asked her. Can they give us two yellow, and we have to figure out what's glucose in the line.
Oh, that's.
For Excel for the last.
I'll tell you guys that right now. Yes.
So we only need to know number 2?
No, you need to know all of them.
Oh, okay.
but
I'm
we-
saying number 2 is only lactose and glucose. That's all you need to worry about. for the phenol red. Phenol red. This is the one for the urease test. Urease test. Urease test.
So it starts,
But if it stays yellow, then it's fermenting.
It stays yellow, it's
No,
different.
it's really
And that's that neon pink, right?
That's.
Also-
because it starts off yellow. And if it changes. Oh, so it'll turn the medium pink under alkaline.
Yeah, yellow is the original color. It says right here. The slant will remain yellow, its original color, and will become more yellow due to the production of acid. So it starts yellow. It says it right here. Right here. You guys need to stop confusing me. Stop talking and confusing me.
There's contamination and it turned red, you would know there's contamination.
Yeah. So it starts yellow. If it stays yellow and becomes more yellow, it's acidic. If it becomes red, it's neutral. And if it becomes pink, it's breaking down the protein or the urease. Yeah, the urea. Urea.
And then, so it starts fermenting for yellow.
So it's already acidic, right? The
Mm.
initial, stuff.
We could take out the fermented of the sugar part. It's just acidic. Yeah. And then this one, which one do you guys think it is? Yes. Citrate. So what does citrate break down?
I would ask them how I did.
I wanted to fill up all of them so we can see. We can explain it. I don't need to do it. And I read it.
The same one is the one that's in this packet. And it's ABCD, so this one is ABCD.
Yeah, that's true. Well, that's the point. If you missed it, there's something that I don't know.
Oh, yeah.
Restoration or something.
Okay.
Arrow. Next year. That says as only carbon source?
Yeah.
So it did. turn 50%. Yeah.
So, just in case she asks, I'm not gonna explain the process because I don't want to confuse you guys even more. Before the carbon fermentation, for like a free-for-all question, a written question or something, just in case she asks, because this was on my test, just know this, that the bacteria breaks down glucose, okay? No, I said for the fermentation. It breaks down the glucose, which then uses the pyruvate which then the pyruvate becomes acid or gases, which causes then the pH to change, which then causes the color change. She might just ask like, what's the process of how you get a different color, or how that occurs. That's how it occurs. I'm not gonna go through the whole glycolysis and all that. You guys went over that in chapter 3, chapter 4. So if you guys are confused on that, go back to that chapter. So it's glucose. So bacteria breaks down glucose, which then makes the pyruvates. Then the pyruvates forms into acid or gases, which then that changes the tube color because of the pH change.
Right, thank you.
You're welcome. Skip all that because you guys don't need to know this. All right, so both broths contain glucose, lactose, and sucrose. Okay, so they all contain a sugar. They all contain a peptone, which is a protein or amino acid. So if the bacteria does not break down the sugar, they may use the peptone instead. Okay. Yeah, the BCP broths. Huh? Just 2.
Both process, so it's only the BCP?
Yeah, only BCP.
Why did we say both broths? So just,
There's glucose and lactose, yeah. It also has a pH indicator, which that's the one that changes colors. And they also contain, this is your key point when determining if it's a lactose or glucose tube and not something else, okay? This is your key determination right here, so please pay attention. The broths will only contain a Durham tube. Nothing else will have a Durham tube in there. Nothing else. BCP.
Yeah.
Yeah. But what's actually?
Durham tube. No, you're good.
This is to find out glucose,
No, this is the one that catches bubbles.
but they're only in glucose.
Only in BCP, which is the glucose and lactose broths. BCP. Brian.
Like differentiate, have you differentiate like a test tube?
Yes,
Yeah.
Oh,
So like-
for sure. 100%. Oh yeah.
yeah.
And glucose. They're in both. Exactly like that. Listen to me, love. I can't say that. Repeat it louder.
Be a salt broth tube, and then one of these tubes will both be yellow, so you know that it's
Oh,
BCP, it's a kind of Durham.
yeah, okay.
Spot.
And she said she'll accept glucose or lactose as an answer.
Okay, yeah, see, so that just answers your question right there. She's not gonna have a glucose and a lactose. She's gonna have one, and you have to determine it. When did she say that? This morning. Okay. Very good.
and you will know that it had fermented either glucose or lactose due to the color change?
Yes. Okay, so the Durham tube will be the one that tells you if it's gas or not. Um. Okay, are we good with this? This is the carbohydrate fermentation one. You guys good with that? Yeah? Okay.
That covered urease, citrate, and fermentation of glucose. and
Well, I
fructose.
just covered carbohydrate fermentation. The pH indicators I went over because I wanted to put them all together, but I haven't gone over the other ones yet. All right, moving on. E and B. What stands for? Eosin, methylene, blue. So, do you guys remember, is this selective or differential? Anybody? Selective? Wrong, it's both. It's both. You need to know it's both. Huh? So, the selective part of it is pretty much just asking who is allowed in. So E and B contains eosin Y and methylene blue. Both of these are dyes. And these dyes inhibit gram-positive bacteria. What does that mean?
Only gram negative.
Right. So if you have no or very poor growth, is it gram positive or gram negative if you grew it on this agar? Gram positive, very good. Because the dyes inhibit gram-positive bacteria. If you have good growth, what does that mean automatically just by looking at the agar? That it's a gram negative. So it will only gram negatives will grow on an E and B. So if you put Staphylococcus aureus on there, is it going to grow? No. Now, the differential part of the agar is basically asking, what is it doing? What is this bacteria doing? Okay. So they're asking, are they fermenting lactose? Fermenting lactose or sucrose. Both of these sugars are on this agar, lactose or sucrose. It's not and, it's or. Or. So more acidic, you get a darker color because the acid mixes with the dyes. If you get colorless, that means no fermentation happened. So bacteria did not ferment the lactose or the sucrose, okay? No acid was produced. No acid produced.
The differential, whether or not it's fermenting lactose or sucrose, is going to be an indicator of what kind of bacteria it is as well? Does it matter if it's?
Doesn't matter if it's lactose or sucrose, but if she asks what sugar, so if she throws in there glucose as part of EMB, what are you gonna say? Wrong, right? Because it's only what? Very good.
Darker color is the, colonies we see, not the active color, right?
Right. Um,
So for the EMB, the only colors that I saw was pink, purple. and
I'm
green
getting to
color.
that. Not done. So then we have pink or like a light pinkish, purplish. Nope, this is still part of 2. And these are the colonies. So that means that some weak fermentation happened. So that means that there's some acid produced, not a whole lot. And these are called non-fecal coliforms. Now, if you have a dark purple or black colonies, that's a strong fermentation. And that means more acid was produced. And if you have a green metallic sheen, that's automatically E. coli. That's it. That's all you need to know. Well, I'll give you guys the detail just in case, but that's the bacteria is E. coli. That means it's extreme lactose fermentation happened. Huge acid production. Okay.
The other ones is just spell out, right?
Mhm. Yeah.
It's the underline.
So,
Whole
Whole
equal.
Or any case stays right, make sure it's agenda and PC. And make sure you guys underline.
All right. So if I have a colorless colony, what does that mean? A colorless colony, what does that mean? No fermentation. If I have a dark purple, what does that mean? If I have a pink and purplish, if I have green, extreme, extreme. What else can be on there besides the colors and the colonies? You guys remember?
Like the shape of it.
Starts with an M. No, that's a whole different agar.
Yeah, that's a question one more time, please.
What else is on the agar when you open it? What else do you have to pay attention to besides the colors? Huh?
The mix of
So
it,
you put
it's like
So
they-
mucus?
Mucoid, which is the slimy, shiny,
Wasn't it only,
gooey
that happened in Italy?
colonies. No,
I think something else had.
this is due to capsule production. And which bacteria had this, Alvia? Yes. Pterobacter, which is also known as this one's they can, but it's often seen in Enterobacter. So if you see it, it's most likely Enterobacter. Also, it got reclassified, so it's no longer called Enterobacter, in case that's a bonus question. So that's its new name.
You said it's a bonus question?
I said if it's a bonus question. I haven't seen the test. She hasn't made it yet. Yes.
After metallic green, you can score.
Shane. Mhm. So I'm gonna give you guys 3 plates.
So for the selected, we've already basically touched on all bases.
Yeah.
Is there examples?
I'm just writing it out, see if you guys are paying attention. So we got plate A, plate B, plate C.
Plate C is equal.
Coli, very good. Anything else for plate C? No, very good. Plate A and B. Very good. So the ones we have that are gram negatives, non-fermenters, are Pseudomonas, whoops, aeruginosa. We also have Protus. What was your question?
You think if this was a question, like we would have to know what the bacteria were?
Yeah.
Or we would just say them?
I do remember there's one that she's gonna give you. I think there's 1 or 2 where she gives you the entire test, the agars, the broths, and everything, and she says figure it out.
She said everyone did very well on this.
We did.
So you have to
We did.
memorize.
That was my
Did
high
she-
score. I got like a 102. Yeah.
So you pretty much have to memorize this whole sheet.
She gave us one too.
She gave you guys one, like a given one. So then maybe study that one. And then we have Proteus vulgaris. I mean, we did have -2 because we didn't do the BSLs or BSLs. The BSL2s. But yeah. So plate B, that gives you Serratia marcescens. Vitro who? Me? No.
Are you trying to be?
Yeah. Grundy. And plate C, just E. coli. Just E. coli. And then just so you guys know, the Enterobacter, I don't have this on here. Yeah. Yeah, so say she gives one that is, say she puts the green one on there. That's the green one. You have to figure it out, right? Automatically, you should know off the bat that's E. coli. You don't even need to look at any other tests, E. coli. If she puts something with good growth and it's like pinkish colonies, it's like pink, light pink, then you can eliminate E. coli. You can eliminate the Pseudomonas and the Proteus. All you have to worry is about Serratia and Citrobacter. And the Citrobacter, you would look at which test? Mm-hmm. And then that one would be blue,
What test?
the citrate, and then you would narrow it down between, I think I think it would be the Urus test, or I'd have to look, I have to double check, but
One test will rule out both. So this test will rule out E. coli and oxidase will rule out Pseudomonas
Mhm.
Isn't she gonna give us the economist key for this one?
I don't think so.
What do you mean by she's gonna make us like eliminate what we? I would have thought that was.
I think you're.
Oh, you guys are gonna have one?
I said, heard some, she mentioned she gonna give us the economist key. And we have to memorize on the results, not the bacteria and they are
giving us an economist key, like we have to do one for our physics exam. So I wish she gave us another. She didn't give you guys much to give us.
For telling someone that she gonna give us the economist key. Like right after that lab with biochemical test, she gonna, I'll give you the economist key and you're gonna have to figure out what the. Based on the result, don't memorize result, memorize the result. That's how she said it. And she gave us that handout page.
Didn't she say for the dichotomies, you gotta use something to explain for the? Yes.
No, she's saying about how it's gonna.
Yeah, see, I don't know. The only thing I have is the list that she's sending to the office to have to set up for you guys. So that's all I could see. I can't see if there's a dichotomous key or not.
But I heard
Like
her say
how,
that.
like you're telling us to eliminate right now, it would be like.
She said it's crazy to memorize each bacteria and what the test results were. Just memorize which color change. And I'll give you the economist key and you test and then based on those results,